Neurons and supporting cells are the type of cells found in the nervous system. Let's make a brief review of anatomy for the nervous system. Nervous system is one of the 11 systems in our body, crucial in our internal physiology, and is composed by two main branches: the central nervous system, composed by the brain and spinal cord, and the peripheral nervous system that is composed by cranial and spinal nerves that are emerging from the brain or the spinal cord and produce innervation in the rest of the body. Now, in this tissue, we found two different cells: the neurons and the neuroglias, or also called glias. The neurons has the function to just conduct impulses or electrical activity. And in general, 99% of the cases, the neurons cannot be divided. They cannot be reproduced. If the neuron died, that cells is not possible to be reproduced. The neuroglias are the one, and there are different types, that the one that support the neuron activity. In other words, are the one in charge to create the external environment of the neurons optimal in order to produce that electrical conduction pure and clear, and they can divide. As a general idea of the neuron functions, we can mention that, obviously, we have different type of neurons as well, but they are in charge to conduct electrical activity. They conduct electrochemical impulses. They are also used to produce chemical regulation through the neurotransmitters, are capable to sense change in the internal environment or the external environment as well, because part of the neurons could be sensors. But the variety of functions is really very broad, very complex, that you can read in this slide, that are in charge for learning, to produce a memory, to control muscles and glands. The really variety of functions is very, very broad and very complex. And also, as I mentioned before, they cannot be divided. They can be repaired eventually, but not divided. To be consistent with anatomy, it's important for you to identify the different areas in the neuron, like they are the body, the dendrite, the axon. Doesn't mention in this slide, but the axon terminal is the other part of the neurons. So the bodies, the neuron body is the one that contains certain organelles, obviously the nucleus, and very important, this area, the body, is the one that receives information coming from the dendrites and is in charge to send information to the axon. The dendrites are the ones that are like a little terminals, like a network around the dendrites, and they are also in charge to conduct graded impulses toward the cell body. Keep in mind about this word, graded, but by now it's important for you to know that the dendrites are, as well as the body, are capable to receive or establish synapses with other neurons, and they receive electrical information from that electrical impulses. That electrical information is captured by the dendrites and sent it to the body. When the body receives that electrical information, is also conducted to the hillock. The hillock is the area in the neurons that connect the body with the axon terminal, or the axon, sorry. The axons are the ones that conduct action potentials. Action potentials. Within, from the dendrites to the hillock or the body to the hillocks, the conducting electrical activity is produced in a way that is graded impulses. You don't have to know right now exactly the difference, but be familiar with these two terminologies, graded impulses and action potentials. The graphic obviously represents the image of the neuron with the different parts, the dendrites, the body, the nucleus inside the body, the hillock, that is the area that connects the axon with the body, the axon by itself, and we cannot visualize the axon terminal. Another thing that you can visualize are the blocks of myelin in the axon, and they are separated by a structure or an empty area of myelin that are called nodes of Ranvier. It's important to identify the action of a myelin. When the axon is wrapped by myelin, the conduction of that electrical impulse is faster than when the axon doesn't have myelin or wraps on myelin. Another important information about the myelin and the axon is the accumulation of axons with myelin are the one that determine the different areas in the central nervous system, right? Either the brain or the spinal cord, that are the white matter and the gray matter. In the gray matter, regardless if we are talking about the spinal cord or the brain, the gray matter is a high concentration of neuron bodies and dendrites, where you have really high amount of synapses with other neurons. When you have a white matter, either in the brain or the spinal cord, is a concentration of axons with myelin, because myelin has a particular more clear color than the neurons by itself. We have different classification of neurons based on different characteristics, but the one that I want to emphasize is the three types of neurons that we talk at the beginning of the semester: the sensory neuron, the motor neurons, and the association or interneurons. The sensory neurons sensory and motor neurons are part of the peripheral nervous system. Sensory neurons are the ones that are coming from the sensors, the one that capture that change in the value, whatever value could be body temperature, pressure, blood pressure, could be glucose level, could be a different kind of osmolarities, could be hormone levels, the list is unfinished. So these sensory neurons are the ones that send the information to the central nervous system, are called sensory neurons or afferent neurons. The motor neurons are the ones that take the action that need to take place to adjust the modification in that value out of the central nervous system and reach the effectors. These motor neurons are the ones that reach the effectors. The other neurons are the interneurons, that, by the way, are the largest amount by far of type of neurons, way more than sensory, way more than motor neurons, interneurons, and they are always located in the central nervous system. Central nervous system, remember, could be the brain or the spinal cord. I try to recreate the graphic I draw in the whiteboard at the beginning of the semester, and you have one particular video that explains this topic in more detail. But I try to recreate it in a PowerPoint, and I really apologize if it doesn't look very pretty, but it's the way how to visualize that famous drawing that I prepared at the beginning of the semester. But basically, the central nervous system is represented in that yellow color in the center of the graphic. Central nervous system is composed by brain and spinal cord. Now, we have the sensors, because it's kind of a loop, right? The one that is used for the famous negative feedback in order to control the hemostasis. But the sensors that are on the left side of the slide, the sensors are the ones that detect the deviation or the modification of the normal value. That are the function of the sensors, and sensors are the one that pass the information to the sensory neuron. The sensory neuron is also called afferent neuron, and this afferent or sensory neuron is the one that takes that information in a way of the electrical impulse and send the information, take that information into the central nervous system. When it's in the central nervous system, communicate through the nerve impulse, communicate with the interneuron. The interneuron is the one that has the function to decode, to analyze what is the information, and based on this information, send the action that need to take place. Interneurons are always in the central nervous system. They cannot leave the central nervous system. Therefore, they need to use a kind of a messenger, and the messenger is the motor neuron. The motor neuron is the one that takes the order and reach the effectors in order to adjust the value to bring it back to normal. Now, that motor neuron or efferent neuron, has two branches. Could be autonomic motor neuron or somatic motor neuron. The somatic motor neuron is the one that reaches voluntary effectors, only voluntary effectors. That voluntary effectors are always skeletal muscles. Therefore, the somatic motor neuron is the one that reaches the skeletal muscles to adjust the value. Keep in mind that the effectors is not only one, could be, and in general it is, more than one. The autonomic motor neuron is the one that is using one or the other branches of the autonomic, that is sympathetic or parasympathetic nervous system. Both are opposite between each other, but the combination between these two are the ones that are the key to keep our internal environment in balance. And these effectors that are reached by the autonomic motor neuron are always involuntary. Could be organ, glands, vessels, you name it, but they are always involuntary. As I mentioned before, the somatic motor neuron is the one that is used to reach voluntary effectors, like produce voluntary control, like the skeletal muscles, and they are responsible for reflexes as well. On the contrary, the autonomic motor neuron is the one that innervate involuntary effectors. Effectors or targets are the same, like smooth muscle, cardiac muscle, glands, vessels, and the list could be even larger. And use the following branches. I mentioned that is the sympathetic or the parasympathetic. This graphic represents exactly the same graphic I presented before. The only thing I want to mention is the sensory neuron is only one neuron. The somatic motor neuron is always one neuron. The autonomic neuron is not one neuron. The autonomic nervous system, either the sympathetic or the parasympathetic, always use two neurons. The one that is emerging from the central nervous system, that is the preganglionic, and the one that reach the effector that is the postganglionic. I will develop this topic in more detail when is the time to learn about the autonomic nervous system. Now, to talk about the neuroglias, we have certainly neuroglias in the central nervous system and some of them in the peripheric nervous system. The ones located in the central nervous system are the oligodendrocytes, microglias, astrocytes, and ependymal cells. Now, the oligodendrocytes are the one in the central nervous system, are the one that produce myelin. And characteristic that one oligodendrocyte, that is a cell producing myelin, is producing myelin for several neurons around, axons around. The microglias are the one that are around the central nervous system, but they have a tremendous importance because it's a type of white blood cell. The function is to phagocyte a foreign molecule that doesn't belong to the central nervous system, including pathogen if there is any. So the function of these microglias are crucial to keep the central nervous system extracellular fluid clean of any particle that can interfere with the neural function. The ependymal cells, and I want to leave the astrocyte for the last time, the ependymal cells are the ones that are always close to the ventricles in the brain and are the one in charge to produce the cerebrospinal fluid with all the functions of the cerebrospinal fluid. But the ependymal cells are the only one who produce the cerebrospinal fluid. The cerebrospinal fluid has the function of protection, but also because it's kind of protecting all the central nervous system outside, so the brain and spinal cord, they are floating in that cerebrospinal fluid, but they are also cerebrospinal fluid inside the brain and inside the spinal cord that is in between the meninges, if you remember, and in the brain. In the ventricles, we have 4 ventricles in the brain. The function of this cerebrospinal fluid is not only protection, it's also absolutely necessary to send the nutrients to all the cells in the central nervous system, but also is used to pick up the garbage molecules that these neurons and neuroglias produce and remove it from the central nervous system. Finally, the astrocytes. The astrocytes has a very important function, but is regulate the external environment of the neurons. And I will use one particular slide to describe the really large variety of function of the astrocytes by itself. Mention that we have neuroglias in the peripheric nervous system. There are 2 types of them. One are the Schwann cells that are similar like the oligodendrocytes. The Schwann cells are producing myelin as well in the peripheric neurons and always in the axons. And finally, the satellite cells that are located in the ganglionic areas of the peripheric nervous system and support the cell bodies within the ganglia of the peripheric nervous system. So support the cell bodies because it's in the ganglia of the peripheric nervous system where there is a synapse between the preganglionic and the postganglionic. This graphic clearly represents the type of neuroglias found in the central nervous system. Pay attention about the astrocytes that are like, they have like an extended feet. One of them are located around the capillaries nearby, and the other one are around the dendrites, cell bodies, and they are also around the neuron terminals. So please visualize the oligodendrocytes, the microglias, and also the ependymals right here. Yes, The myelin is produced by two types of neuroglias. On the left side, you have the Schwann cell that produce myelin in the neurons of the peripheral nervous system, and the oligodendrocytes that produce myelin around the axons of the central nervous system neurons. Yes, astrocytes need an special attention, not only because they are the most abundant glial cells, also because, as I mentioned before, they have extended or ending feet associated with the blood capillaries nearby and axon terminals, but they are crucial in how they keep the external environment of the neurons very clear for a better function of them. Please pay attention in this graphic the location of the astrocytes and how the astrocyte keep the extended feet around the neuron terminal, around the axons, around also you cannot see over here around the bodies, dendrites, but also around the capillaries nearby, because everything has a reason for, and I'm going to explain in the next slide. So, the functions of the astrocytes are several, and all of them are important. One of them is, the function of them is to take the excess of potassium from the extracellular environment to maintain a balance of this ion. Potassium is mainly kept or in large amount inside the cells. The neuron is not the exception. So when we have excess of potassium outside the neuron, interfere with the electrical conduction inside the neuron. Another function of the astrocyte is at the neuron terminal, has the capability to absorb the extra amount of neurotransmitter that is not needed to pass the electrical impulse to the other neuron. It's important to keep in mind that the neurotransmitter, whatever neurotransmitter is used in the central or peripheral nervous system, in this case we are talking about the central nervous system, because astrocytes are found only in the central nervous system. So, because of that, the excess of neurotransmitter need to be removed when it's not needed. And one of the way to remove it is taken by the astrocyte. There are another ways to remove it, but the astrocyte has a crucial importance to remove or absorb the excess of neurotransmitter that was not possible to be dissolved or disintegrated chemically in the cleft. The other function of the astrocytes is they have the capability, one important thing is they have the capability to take or absorb glucose from the blood, from the capillaries. They have the capability to take glucose, and they keep the glucose inside the astrocyte because they have the capability to store the glucose as glycogen, very unique characteristic. That glycogen is used to either be break down into the glucose molecules that are going to be used by the neurons nearby, because neurons cannot store glucose. They need glucose all the time, and from all the cells in our body, the neurons are the one in largest amount of need for glucose. But also the astrocytes produce lactate that is used by the neurons to produce energy. Now also very important, the astrocytes are in charge to regulate the neurogenesis, that is the repair of the neuron in case the neuron is damaged. But is highlighted in red that one crucial function of the astrocyte is produce blood-brain barrier, BBB. What is blood-brain barrier? Let's take a look about the capillaries, specifically the capillaries in the brain. The capillaries in the brain, even when they are considered continuous capillaries, they are special because the gap in between the simple squamous epithelial, they are not like any other continuous capillaries. In any other continuous capillaries, that gap is something that exists. In the brain capillaries, that gap is almost inexistent. It's almost the simple squamous epithelial cells are packed between each other, and this one has the function. They have very tight tight junctions by which these simple squamous epithelial cells they kept very close between each other, and the function is for protection. We don't want to allow too much filtration from these capillaries into the extracellular space between the neurons, because we want to keep that external environment of the neurons as clear as possible. So the substance can only move from the capillaries by very selective process of diffusion through the endothelial cells. Diffusion, active transportation, and bulk transport. However, this action is very selective due to there is no filtration. Pay attention that there is no filtration. There is a diffusion, very selective, active transportation or bulk transport. Now, this one is possible because the shape, the specific shape of that capillaries, and also sorry, also because the astrocytes that keep that particular extended feed around the capillaries that is blocking the filtration to any molecules. In other words, the astrocyte is the one that is helping to that selective process. Now, also, the astrocytes influence in the production of ion channels and enzymes that can destroy toxic substance by secreting glial-derived neurotrophic factor. And very important, that action, blood-brain barrier, that is in combination between the specific capillaries in the brain and the astrocyte extended feed, create a conflict with medication that the patient might need to try different kind of disorders, kind of Alzheimer or Parkinson or meningitis or bipolarity or depression, any kind of medication that need to reach the central nervous tissue, central nervous system neurons is interfere with the action of the astrocytes. And it's one of the reasons why when the patient needs a medication for any kind of the disorders I just mentioned, and there are obviously way more that affect the central nervous system, that medication takes a while to take effect, because probably the patient needs to wait minimum 3 weeks to see if the medication is working. So keep this one in mind because it's a very important information about the blood-brain barrier. In one way, it's designed to protect the central nervous system tissue, but in another way, interfere with medication if the patient is affected by one of the kind of disorder that I mentioned.